And we do move now to the next presentation by Cristina Fossi.
Thank you very much, Professor Skoulos, and thank you very much, Francesca Marcato, for the very nice and kind words for our project. And we really think that the message that you give us in terms of transferring the methodology in other areas of the Mediterranean Sea will be reached from our team in a very positive way. And today, I think we start at this section, we start this very comprehensive evaluation of the main result that we obtain in the testing phase. There was something strange in the sound. In the testing phase of the monitoring activity in the former marine protected area. And I have the pleasure to present the result that we have obtained during this final 2 years of activity in the monitoring of the large spammy Pelagos Sanctuary. Obviously, I will be present the result, but as you can see, a very large team of researchers of several institutions have given a very important contribution of this monitoring phase. Probably you know the Pelagos Sanctuary, what is. The Pelagos Sanctuary is not really a marine protected area, but it's something probably more extensive and important in terms of biodiversity, is considered as a SPAMISH, so special protected area of Mediterranean importance. And probably you know the Pelagos Sanctuary as the marine mammal sanctuary in the Mediterranean Sea that was a protected area that coming from an agreement from Italy, Monaco, and France from the protection of marine mammals. So this area is one of the hotspots of biodiversity in the Mediterranean Sea. So it's extremely important, but in the same time, again, it's one of the areas more exposed to the threats of human activity, and especially the cetaceans that are the charismatic species that live in this area are undergo to several impacts. The first one are represent from direct impact, such as bycatch, noise, ship strike, but the most difficult to study and identify are the indirect impact that represent sublethal effect from the biodiversity, and one of these indirect impact are represent from marine litter, including microplastic. So in this project, our main goal was try to define the impact of macro and microplastic on the Mediterranean biodiversity living in the Pelagos Sanctuary, starting from mussels, but focusing particularly on cetacean species. Just briefly, which kind of experimental design we adopt in order to monitor so wide area. Obviously, the approach that we have to use to monitor a so large pelagic area, it needs to be customized to the environment. And the approach that we have used, I'm sorry that is the color are not very bright, but on the left side, you can see that one of the most important approach that we apply is using a marine litter hotspot identification. So in collaboration with the Consorzio Lamma in Italy, we developed for the first time a model of concentration of macro and microplastic in the Pelagos Sanctuary. As you can see, the red area are the potential hotspot and the blue area, the cold spot. So our first directive was orientate the monitoring in the direction of hotspot and cold spot, but but also, very important, to focus the attention on the area in which the biodiversity are more frequent, and particularly we move to define the core distribution area of cetacean in order to see if there is overlapping between hotspot of marine litter and the presence of cetacean. The monitoring activity was carried out in May-September 2019. We have more than 34 days of sampling, more than 2,000 miles monitored, 34 researchers and 8 institutions provide the information that I will show you. Here again, we don't see very much, but one of the most important novelty that we apply in this project, especially to monitor pelagic area, was to simultaneous monitoring the floating litter, microplastic and biota in the area that we analyze. And here you can see that at the end of the sampling activity, we reach a very large number of samples. We have obtained more than 140 superficial microplastic sampling, 273 floating litter. But the most important thing that during all the campaign, we have the biota sightseeing. So in order to see where there is the hotspot or litter, we have the information of the presence of species as well. And in the same time, we are able to collect for the first time neostonic bioindicator, as we show later on, and cetacean sample with the non-lethal technique of the skin biopsy procedure. Just briefly to move off the main result. I don't want to spend time in methods because it's important to see what result we achieve. The first data is to have the pictures of the concentration of macro and microplastic in the study area. And here is important to see, and my colleague Cristina Fossi will show later on, that two of the area that we monitor, the Pelagos Sanctuary and the Toscan National Archipelago, are strongly interlinked because the Toscan archipelago is inside the Pelagos Sanctuary. So one of the goals is to see if there is different of concentration of the different plastic presence in the two areas. And as you can see here, concerning microliter, the concentration in the Toscan archipelago are higher than in the rest of the Pelagos. So it's 0.36 compared to 0.18. And the same trend was reported also for the macroplastic microliter. Again, we have a very strong difference between macro, the Pelagos and PNAT. So PNAT appear a kind of more hotspot inside the Pelagos. But on methodology result that is very important to show also in terms of implementing marine strategy and IMAP, we found a very high correlation between the two parameters. So we found a very high correlation between microplastic concentration and floating litter with a wide range of data. That is very important information that can be served as supporting the model distribution data. The model distribution in the Pelagos Sanctuary confirm in a very clear way that the theoretical area of hotspot and overlap with the field data. So that in other way, the field data that we obtain completely confirm the presence of hotspot in some area of the Pelagos Sanctuary. And obviously, what is very important for us is to link what happened in the environment and the potential impact on biodiversity. And obviously, the first focus was given to the charismatic species that living in this area. So the cetacean living have reproductive activity and feeding, especially in the summertime, in this hotspot area of plastic concentration. And in this project, we developed for the first time several new techniques to define the impact of marine litter on cetacean. And in particular, as we mentioned before, we applied this threefold monitoring approach using both stranded organisms that you see on the left side of the bottom and free-ranging organisms. So these two kinds of sample can allow to define the presence of ingestion of marine litter, but also define the transferring of contaminants to the litter, to the tissues, with the detection of plastic additive, and finally, to define the toxicological risk with the presence of some specific biochemical endpoint that we define biomarker or omics techniques. So as you see, as you know, one of the techniques that we mostly apply for the study free-ranging organisms are the skin biopsy, and later on you can see the movie that Marco or the other colleague of ISPRA have realized during the campaign. So I don't want to boring you with this information now, you can see later on. And but what is most important that in this project, for the first time, we develop some new diagnostic technique to define the presence of plastic additive and particularly the responses of this organism to pollutant. Here we have some of the skin biopsy that we collect in the Pelagos Sanctuary with the fin whale, sperm whale, striped dolphin, and bottlenose dolphin. Plus with the colleague of Cabrera, we are able to collect other sample of sperm whale, tursiops that can be used in order to compare the concentration in different areas. But what is very important to underline, as you remember from the presentation of this morning, some of the species, in particular fin whale, can be used as a potential indicator of the impact of marine litter in the entire basin. It's a wide range bioindicator species. And I just say this in order to see the result. Because as mentioned before, we have developed for the first time a new technique that is the metabolomics. So we measure the complete metabolomic responses in the skin biopsy of fin whale. And what we obtain in collaboration with this French colleague of Metatool, applying this NMR analysis in the skin biopsy of striped dolphin and fin whale, we are able to define for the first time the metabolomic profile of these animals in striped dolphin. I'm sorry that we can see very well, but you can believe me, that is a statistical difference in terms of metabolomic profile between the fin whale of the Pelagos Sanctuary that are on the right side, on the left side, and the fin whale collect in a defined control area that is the Sea of Cortex in Mexico. So you can see there is a dramatic difference in terms of metabolomic omic responses between the two populations. And what is extremely important to see the relationship between these biological responses of the organism with the plastic additive that we know can be used as a tracers of the presence of litters in the area. And what is extremely interesting to see that again, we found a very strong difference of the plastic additive profile between the population living in the Mediterranean Sea and the other living in the Mexican Sea. So that is confirm our hypothesis that fin whale can be used as integrate bioindicator of the presence and the impact in the entire basin. That is a very novel concept, and I think we are very believe that is very important to stress. So obviously here we come in detail how it's important to use these new techniques in order to define the health status of this organism. This technique can be applied also for sperm whale, for example, collect from the friend of Caprera, compare with the Atlantic one. So definitely that is a very important output. But what is extremely important to see, if you use, for example, the concentration of phthalate and you merge with the hotspot of marine litter in the Pelagos Sanctuary, we found for several specimens a very good intercorrelation between the presence of marine litter in the area and the responses in terms of phthalate concentration. And here again, we have defined a kind of risk map of the concentration of marine litter in the Pelagos and the overlap with the concentration of plastic additive. So that is, I think, a very important outcome of the project. But obviously in the Pelagos Sanctuary, there are not only whales. There are a lot of biodiversity. And we have focused also from the very large charismatic organism to very small bioindicator, such as these species that you know probably is Velella velella. And that species can be very interesting because it can be proposed as bioindicator of the presence of marine litter on the sea surface. So that can be an eustonic bioindicator. And here again, we have developed in our lab, and here I have to mention a lot of people, Matteo, Matteino, Matteone, a lot of guys that work with us, that have worked a lot in the lab. We have developed a technique to detect the ingestion of marine litter in Velella velella, and hence again, what you can see on the right side, we have an overlapping between the hotspot of microplastic and the ingestion of microplastic, particularly microfibers in these species. And so again, a lot of sample was analyzed, but I think Cristina Panti will describe in a more extensive way later on. So the commercial species in the Pelagos Sanctuary in Prat can analyze. What I would like to say that, and I will ask maybe to Caterina to stress again this point later on, we have done a very nice intercalibration exercise inside the consortium in order to validate the methodology. And hence again, we showed that several species such as bob and sardina can be used as bioindicator for commercial species. But one of the great novelty of the project is to apply on the same species, that is the striped red mullet, the threefold monitoring approach. So on the same species collect in the different area, we can analyze the ingestion, we can analyze the plastic additive and the biomarker responses. And here for the first time, we obtain a very nice analysis of all these parameters in which we see that the different areas are completely different on the toxicological point of view in response to the different level of contaminants. But we can stay here for hours describing the result. I don't want boring you. Obviously, one of the target species is caretta caretta, both in terms of stranded, but also on hospitalized sea turtles. And here we show the data that in particular Ilaria have worked very much, both in terms of contaminants and in terms of biomarker. Again, we don't see very well, but you can believe me that the turtles with the plastic ingested and the plastic presence in the feces have a high level of plastic additive and high level of biomarker responses. So that means that we cannot stop only on stranded organisms, but we have to work also on hospitalized sea turtles, and I think is a very important outcome. And again, the hospitalized organism, you can not only collect blood or skin biopsy, but you can easily collect feces. Feces is very easy to collect, and hence again we can see that some there is different of plastic between species present in the hospitalized rescue center, and the specimen with litter have different concentration of some endpoints, such as porphyrin, respect to the species without plastic. So again, it's very important to stress this. And finally, the marine mammals, they are not only free-ranging, but you can find stranded marine mammals, stranded cetacean along the Pelagos Sanctuary. And here what we can do, we have developed a kind of prototype to isolate microplastic from the gastrointestinal tract to cetacean. That is not easy stuff, because you can have meter and meter of gastrointestinal tract in case of sperm whale or other species. And so in collaboration with the University of Padova and the Istituto Zooprofilattico of Turin, we have developed this prototype that you see on the slide. And we have analyzed a huge number of stranded cetacean. And as you can see, the occurrence is 100%. Some species, we have only one, such as fin whale, but another, we have a large number. So all the species analyzed show microplastic. And here you can see the different microplastic that we have found in the different species. And obviously, the fin whale are the top 10 in terms of concentration. And again, we have analyzed the plastic additive, and there is a very high level of plastic additive in the several species living in the Pelagos Sanctuary. But obviously, we have all this team that I showed before. I have worked very much in terms of monitoring marine litter on beaches, that we know is a basic information in order to define specific mitigation action. And here you can see the different concentration of macro litter in beaches in the Pelagos Sanctuary from the Tuscany, Ligurian Sea, and Corsica. And what is very negative to show is that one, that all the beaches analyzed are over the threshold level, several times over the threshold level recommended from the marine strategy. This information, but I forgot to mention before, that is great to the presence of Tommy. We have used probably for the first time in a European project the joint list for marine litter, and that was able to define the sources. That was extremely important, and we show later on how is important in order to orientate mitigation action. Another activity will be present later on from Cristina, because François Galgani, our scientific father, unfortunately is not here with us, and so Cristina will show the result of the hotspot beach monitoring later on Pelagos. And obviously there's again, diagnosis, mitigation, but also diagnosis and define the risk prediction in the area. That is a very important exercise in which, in collaboration with CIMA Foundation, we defined for the first time the sensitive map of marine litter biodiversity and impact on the area, ranging from cetaceans to seabird and turtles. And we saw a lot of overlapping between the concentration of marine litter and the potential risk of the species. So what is, on our point of view, the lesson learned from the Pelagos Sanctuary monitoring? So obviously, when you have to monitor a so large area, you have to try to maximize the effort. So, and the procedure that we use is to have a simultaneous monitoring of marine litter and the presence of biota. We are able to define several hotspot areas in the area. We are able to define that both commercial species and endangered species reveal the ingestion of marine litter, but also we develop new techniques to define the toxicological hazard, and that is final goal is to have the risk prediction in the area and the most exposed species, but also the sources identification that will be an important effort to generate the future mitigation action. So that is the end of my presentation, and obviously I have to give a great thanks to all the people that have strongly contribute to the monitoring activity, both in the area of Pelagos and later on in the PNAT archipelago. And obviously, thank you very much to be here to listen us and our message on Pelagos. Thank you.
Thank you very much, Cristina. I think we are lucky to hear firsthand very impressive results from state-of-the-art innovative research. And I would like to say that combining innovative methodologies with opportunities to have these sensitivity maps that lead to strategies for and design of protection is something extremely useful.